to reach a temperature traditionally, or do they actually stay stable to higher temperatures?
Okay.
I showed the graph, but I kind of missed that.
Yeah, let's see. Can I get back to slide 5 or 6? No. That's, you know, I forgot to mention that when I showed you this data. So, sorry, I guess I have it. All right. So you see that when I heat the blue curve, I get an extra 20 or 25 degrees beyond the ordinary TG before it becomes a liquid. But you're right. Once it's gone up to the supercooled liquid, it's forgotten all of the interesting structure that we provided by vapor deposition. Actually, if it's an OLED display, it's a bad idea to leave it in the sun for long periods of time. They are photochemically degraded by light over long periods of time. All right, let's take a question from the right-hand side. There you go.
Hi, thanks. How does this work change how you interpret experiments with like the confinement effect of glasses? And a lot of those are strongly dependent, obviously, on the substrate that you're confining with. And then I don't know, maybe I missed it. I don't know if you talked about the substrate that you're doing the deposition on. And yeah, thanks.
Okay, great question. So if you're looking, we often are looking at films that are between 50 nanometers and 1,000 nanometers. And in that range, the properties are independent of what the structure is. Okay, and that's consistent with our mechanism, which is all about templating from the top surface and not from the bottom surface. Now, certainly, if you get to really thin layers, you're going to be influenced by the structure of the substrate, and that's something that we're still working on understanding. All right, let's go back over to the left question.
So I'm just curious, let's say I'm running a packing of oranges as possible. Do I drop them one by one on the flatbed truck so that they can
That's a good question. Sort of intermediate state, and they make a good glass, but it's not particularly any better than you would make by standard methods, and that's because colloids really don't have this. We've got 8 orders of magnitude of extra mobility at the free surface compared to the bulk. Maybe in colloids you got 1 order of magnitude or something. But you really don't have this kinetic advantage that we have in these organic systems. So I have to think more about my brain problem. Thank you. All right, well, let's thank your speaker again.
All right, thank you, Paul. It's an honor to be here and to share some of the work we're doing in my group in this field. There are a number of points of contact with probably all of the previous speakers to some extent, certainly with Sharon's talk in terms of entropy as a driving force, actually for self-assembly in cells. So think about biological there's an enormous degree of richness to the set of interactions. There are thousands of different types of molecules, biomolecules like protein and RNA and DNA that are interacting in a crowded environment. And so the interactions, like you see in the simulation on the left, those interactions, despite the enormous number of fluctuations, thermal and likely non-equilibrium fluctuations within the system, those interactions give rise to coherent assemblies, coherent organization. Things like the migrating cell on the top, the leading edge of the migrating cell, or this neutrophil chasing a bacterium in the middle, or peroxide encapsulation in the bottom. So I think it's fair to say that biological systems are the most richly textured form of matter in the whole universe. And if anyone has something more richly textured and can define that, but then let me know. But I think they're going to wait until we find an alien life form and maybe something more highly evolved or something. But that's all in terms of physics. We suspect that there's going to be entirely new physics in biological systems by trying to study the principles that govern self-assembly. So what makes biological systems so interesting, one of the features I think so interesting is that they're a wide span of length scales involved in hierarchical self-assembly. So you can think about at the nanoscale, the building blocks, protein and RNA interactions, let's say, and then somehow those interactions code for organization within the cell, things like organelles, and then the assembly and function of those things that dictate cell behaviors, tissue behaviors, and organ behaviors that ultimately build those tissue robots that you and I. Now, what's interesting, I think, about this is that we actually know a fair amount, certainly not everything, but we know a fair amount about physics that governs self-assembly at the nanometer scale, so the small scale that we're here of biological systems. We can think about structural biology models, protein folding models. We have a pretty good conceptual framework for understanding things at that level. If we go up to the highest length scale, sort of what I'll call the macroscopic scale, we can think about continuum mechanics models. Today is this mesoscale at the sort of micron length scale structures within living cells. And I would argue that we have very little understanding of the rules that govern assembly at that scale. So I'm going to try to tell you about some of the ways we're thinking about that. In my lab and in this field, I'm going to tell you about inspired by soft matter systems that are non-living but have some features, spontaneously at least, that are squishy, they develop fast molecules, things like polymers that can give rise to emergent behaviors, viscoelasticity, and so forth. So we're thinking a lot about the ways what we learn in soft non-living matter and how we can take some of those insights into living cells and ask, can this give us new insights and new knowledge on how cells are organizing their content. are things like processing bodies, signaling assemblies, and an array of these structures inside the nucleus, subcompartments of the nucleus. And in fact, I think it's probably fair to say that most of the bodies inside of cells, most of these kind of organelles that one sees are in this category of membrane-less. So it's not, it's not sort of a small subgroup, but in fact, maybe most of the organization is mediated by these kind of structures. And so I started thinking about these in the context of this problem, which these structures are called T granules, and they're implicated in specifying the lineage of the cell, so the fate of the cell, whether the cell is going to form a neuron or a gonad or something else. And these structures are indicated specifying the reproductive organs of the gonad of a C. elegans worm. And what's interesting is initially after fertilization, you can see that they're distributed throughout the embryo, and green puncta distributed throughout the embryo. But as the embryo starts to polarize, it tends to develop distinct anterior and posterior axis. These structures end up on one side. And when the embryo and so this process, we've learned a lot about how it works. It's mediated by gradients of molecules that are kind of morphogens that control the segregation of T granules into the embryo posterior. What's really fascinating about how this works is that these structures, these T granules, we discovered are liquid-like assemblies of RNA and protein. So despite the name is a granule, they're not little pebbles. They're actually condensed liquids. And you can see that in this movie where we apply shear stress across the tissue in a very kind of low-budget rheology experiment. We just basically squash this tissue and do shear stress. And what you can see is these T granules flow and drip and wet and coalesce with one another. And so this led to a model wherein the segregation of these T granules into the posterior end of the embryo is a kind of phase transition or a liquid-phase separation process, just like what Sharon Glassford was telling us about in a non-biological context. So the idea is that the anterior-posterior axis of this embryo spans the two-phase boundary such that the posterior end of the embryo is in a two-phase region where the condensed liquid states of RNA and protein are stable. And since we first put this model, there's a lot of data supporting this, and it's a very rich problem, and there are many non-equilibrium features which I want to highlight. The cell is very much out of equilibrium. You can see already from the gradient that drives the assembly. So this idea of condensed liquid states inside the cytoplasm and phase transitions driving organization inside cells actually has a long history. We started to discover papers back in the literature as far back as 1899. Pioneering interdisciplinary biologist E.D. Wilson had some of these ideas about the physical nature of the cytoplasm, and he described the protoplasm as a liquid in form of emulsion consisting of these suspended droplets. In fact, we've known that biomolecules can undergo phase transitions for many decades, and the ones who work with structural biology were trying to induce proteins, for example, to crystallize such that you can do X-ray scattering. Phase transitions have also been suggested to potentially play a role in membrane organization, and so there's kind of hints of this in the literature. And it started to become apparent that these kind of phase transitions are ubiquitous within the cytoplasm and the nucleoplasm, the contents of the cell. There's literally dozens of different, or really at this stage, hundreds of papers over the last several years that have come out suggesting that liquid-liquid phase separation is implicated in processes, ranging from mitosis to cell signaling, postsynaptic entities that mediate connectivity in neurons, and essentially almost every biological process that's happening in a cell, there's some connection to the phase separation that was described. So we're really interested in this process, phase separation, and how it is occurring potentially to organize the contents of the nucleus. So the nucleus is where the genome sits, and so we're all familiar from 23andMe or something, with the idea that our genes are dictating phenotype. Something that you may not appreciate is that every one of your cells, every one of her cells has the same genetic code, the same sequence of base pairs that are supposed to dictate the phenotype. And yet she has dozens or hundreds of different types of cells that make up her body. So each one of those cells, its phenotype is supposed to be dictated by the genome, and yet she has totally different cell types, different shapes, different functions. So how does that work? Well, it turns out that it's becoming apparent that these kinds of condensed liquid states are like little dewdrops that are sitting on the grassy genomic landscape, and they're playing roles in mediating expression of genes. And we started to refer to this as a liquid epigenome. So there are many different types of these condensed liquid states which we think are doing this inside cells. Structures like the nucleolus, which I'll tell you about, which controls ribosomal RNA biogenesis, so the protein translational machinery inside the cell. Structures like nuclear speckles, which are regulating splicing, so how genes are being cut, cutting out neutrons, and so forth. And the Hall bodies, which are also playing roles in the regulation of expression of genes. So we started studying this with a postdoc at Webber now at Yale University, and she was able to show that the nucleolus, so this structure that's inside the nucleus, the nomenclature is a bit unfortunate, but the nucleolus, and it assembles around these particular genomic loci that make the ribosomes. And she was able to show some evidence that the assembly of the structure seems to follow a phase boundary. This is we could change the concentration, and the higher the concentration, the more material condensed out of the nucleoplasm. We can also look at the dynamics of the assembly of these droplets and show that there was coarsening of nucleoli around those transcriptional sites that was consistent with sort of classic modeling approaches. So this is the Conn-Hilliard equation, which is a kind of diffusion equation, except it's uphill diffusion. I was going to make a point about how entropy can only drive things downhill in fixed law, but that may be totally wrong now that after Sharon talks, I'll hold my tongue on that. But it's exciting to think about. But generally, one thinks about diffusion down concentration gradient driven by entropy. And so if you want something that's uphill diffusion, you have to have some interactions, something beyond just entropy. And that's what's in that formalism. What's different here, though, is that we have the presence of RNA due to transcription at a particular genomic locus, and that's why the drop in the middle wins out. Another system that we've been using to probe this idea that this liquid phase separation is important in the nucleus, in particular with the structure of the nucleus, is a frog oocyte system. It's really powerful because it's a single cell. It just grows to be very large. It grows to be about a millimeter in size. It's a single cell that you can hold in your hand, and it contains a nucleus that is similarly large. You can also see with the naked eye. And inside that nucleus, there are hundreds of these bodies, hundreds of these nucleoli. nucleus, there's a scaffold, which is an actin network, which is very interesting because it's a nuclear actin network. We're used to thinking actin in the cytoplasm. And that actin network supports these droplets and keeps them all coalescing together. And what Marina found is that if she disrupts the actin network, these droplets actually do all coalesce. What we expected was going to happen was that they would all continue to bump into each other, but what we found is they actually sediment under gravity to the bottom of this nucleus. And that was pretty surprising because I actually been teaching in class how gravity is negligible in cells. And so that inspired me, so that was good. But the calculation you want to do is you want to calculate this central length scale, which is essentially reflects the competition between entropy, which wants to mix up the system, so sort of captured by ET, and the gravitational force, mg, or here expressed in terms of a buoyant mass. And what's interesting here is that the cell has grown so large that it's actually exceeded this gravitational length scale. So that's like the equivalent of climbing a mountain in Bolivia, and you can't breathe, right? The physics is the same. You sort of cross this length scale. And in fact, if you do this experiment, you wait overnight, you do find one single large droplet. And so this is the world record nucleolus. It's about 100 times bigger than most cells, so it's this giant thing. So the other thing that's is really interesting about this work is that Marina found when she was doing these experiments and inducing this coalescence by disrupting the scaffold, she found that there are subcompartments of the nucleolus that actually themselves also coalesce during this large-scale nuclear coalescence. And so I'll show you this movie. The green shows an interior compartment that's characterized by this protein triple-arin, and the red is this outer compartment characterized by protein nucleophosmin. And what you can see is that as these nuclei start to coalesce, the inner core also coalesce. And one has a sense that what you're seeing here is a kind of liquid within a liquid, as you can see in these higher resolution images on the right. And in fact, that's exactly what we think is going on. We were able to show that we could reconstitute this liquid within a liquid assembly in vitro with purified components. What's remarkable about that in vitro reconstitution is that the core shell architecture is maintained. So these green proteins, triple-arin in particular, is on the inside and nucleophosmin is on the outside. And that is known from multi-phase with the higher surface tension with respect to the surrounding fluid will be engulfed by the other droplet to minimize that surface energy cost. So basically, if the high surface tension, surface energy, then if it's engulfed by the other droplet, will that interface will go away and the energy will go away. And so we were able to do some surface contact angle measurements and surface tension measurements to show that that was consistent. It was also consistent with computational work that we did together with Rohit Papu's group at Washington University in St. Louis. So we like thinking about this because there are dozens of different types of these structures, and often they do have subcompartments or there seem to be multiple liquid-like phases, which includes things like the stress granules. So we're interested in this idea that the relative surface tensions again structures the contents of these other bodies. There are other there are other bodies that don't interact at all, so they're not engulfing one's not engulfing the other, but they're not fusing or coalescing or interacting at all. And so we think that reflects a high surface tension that would be associated with the interface between those two. Again, it's known that in multi-phase liquid systems you would have complete exclusion of those droplets. There are other biological systems where there's partial engulfment of the droplets, one partially within the other, which is associated with sort of relatively more equal surface tensions. And things like this beautiful image of this Cajal body and the attached structure called the snurposome, which I would think is such a cute name, especially for something that looks like Mickey Mouse here. But this is a case we think of a partially engulfed liquid. So I haven't said much yet about the molecular driving forces that are at play here. It turns out that many of the proteins that are driving phase separation are referred to as intrinsically disordered proteins. So they're proteins that are conformationally heterogeneous. So I think everybody knows that proteins are linear polymers. They're linear chains of amino acids. But we generally don't think of them as polymers because we think of them as having a compact three-dimensional shape that dictates their function. So it turns out that many proteins are actually conformationally heterogeneous. So rather than being folded into a single compact shape, they're actually sort of fluctuating around. And I'll show in this movie this is a simulation of a protein like this, one of these intrinsically disordered proteins, or IDRs. These are closely related, by the way, to what are referred to as low complexity sequences or prion-like domains. And so we and others in the field over the last few years have been intensely focused on these IDR proteins that contain IDRs because when you purify these proteins and look at them in vitro, what you find is that they readily phase separate into these condensed liquid states, which we think corresponds to the same kind of molecular driving force that the corresponding structure in the living cell. This includes proteins like LAP1, which is one of the important IDR-containing proteins, the P granules, these germline specifying structures that I talked about at the very beginning, and also nucleoli in the bottom two examples. And in this example, the yellow droplet that's from a fungus. So these are just a few examples from our work, but there are really many dozens at this point of intrinsically disordered regions which are thought to drive phase separation. Another key molecular architecture of proteins that are driving phase separation is oligomerization. So a lot of the proteins, and we've just really started to fully appreciate this pretty recently, is that a lot of the proteins have they'll have these intrinsically disordered regions. So this is a linear representation of the domain structure of a protein. You'll see that blue intrinsically disordered region in the middle. On the left is what's commonly seen as an oligomerization domain. So that could be a dimerization domain, so homodimerization, or the protein I already discussed, nucleophosmin, it has a pentamerization domain. And so some sort of self-interaction that can be of that flavor. And then they'll often have a substrate binding domain, which could be an RNA in many cases, and DNA can be as well. And so we started thinking about these architectures where you have oligomerization and intrinsically disordered regions, which are interacting with themselves, we think, in a sort of weak multivalent fashion to drive phase separation. The issue in the recent developments of this field is that there have been very limited tools to study phase separation, particularly in the context of a living cell, embracing the full complexity of the cell. And so my group has been investing in developing technologies to be able to probe and control phase separation in living cells. And And this involves an effort from a number of individuals who are shown here. I want to highlight in particular Youngdae Shin, who's a former postdoc, is now a faculty member at Seoul National University, who really got this work started. It's been continued in next generation versions of this by postdoc Dan Braha, grad student E.J. Chang, and we benefited enormously from collaborating with my colleague Jared Tocher. The idea with these systems is we're taking advantage of optogenetic proteins. So these are proteins typically borrowed from plants that can interact in a light-dependent fashion. So you shine light on these proteins, and something changes about their conformation. They will open up binding pockets or interaction domains. And so the idea was we would have a light-dependent oligomerization. And so I'm going to show you a little bit about some of these systems. The first generation is this octodroplet system, and then we have this correlate system and a cast-drop system. I'll tell you about the latter two. So the correlate system is really powerful. What it is built around a self-assembled scaffold 20 former. We're primarily using this protein self-assembly ferritin. And we've engineered each of those subunits to have one of a light-dependent heterodimerization domain. The other, the light-dependent binding partner, that the other of the heterodimerization domain, we hook up to whatever protein we want to recruit to that core. So the idea is we shine light on the system, and then we get this core that now is decorated with the interacting domains that typically have intrinsic disordered regions. So to show you these cells that are expressing these constructs, we shine light on them, and you see that you get phase separation within the nucleus of these cells, and forming these condensates within the nucleus. You can have a lot of fun with this. This is toggling the light in time and asking questions